Microbes are the future. They are. It's just you can't say no to that, because microbes are just gonna make everything for us in the future, from plastics to fuel to our food. They're just gonna happen. It's just gonna happen. You can't do anything about it. I'm sorry. Can't change it, because they're very efficient at things, and we are figuring out ways to make them even more efficient and better and make new things that they've never made before. And here, microbes convert industrial waste gases into commodity chemicals. So what if we can actually use the byproducts that we don't even use, that's considered waste, and actually use them for useful things? Enter microbiology. Chemicals cost more than just money. Today, petrochemical production spews out 2% of the world's greenhouse gas emissions. Now, researchers have taken an important step to vastly reduce that footprint by using bacteria and waste gases from steel plants rather than petroleum as the starting ingredient for dozens of commodity chemicals. That is such a cool idea. I mean, for chemistry, you can start in a lot of different ways along the process. So this is a way to start with something that's a little bit more bio-friendly. Because researchers may be able to expand it to others, it could help the chemical industry escape its reliance on fossil fuels and effectively remove carbon from the skies. Oh, what a great intro. Man, that's cool. Harnessing biology to utilize waste gas, and produce industrial chemicals is really exciting. A biofuels expert with the Lawrence Berkeley National Laboratory. It goes after two sectors at once that are difficult to decarbonize. Steel production and industrial chemicals. Tackles a very hard problem. Humans have exploited microbes for thousands of years to make products like alcohol, cheese, and yogurt. It's true. Alcohol, cheese, yogurt, it's just food that microbes have already eaten, and we're eating the microbe waste. That's what alcohol is. That's what cheese and yogurt is. They've already grown in there, and we're eating it. We're pre-digesting our food with microbes. Blows my mind. But industrial scale biotechnology didn't really take off until the beginning of the 1900s, when, for example, chemist Chaim Weizmann engineered several species of Clostridium and bacteria to convert starches and sugars into acetone, an essential ingredient for making cordite, a smokeless alternative to gunpowder. Weizmann's success was a key to the Allied victory in World War I and gained him fame. A Zionist politician as well as a chemist, Weizmann eventually became Israel's first president in 1949. Wow, look at that. I had no idea that Israel's first president was a scientist. That's really cool. With the rise of petroleum industry in the 1950s and 1960s, it gave chemical manufacturers cheaper starting materials for dozens of commodity chemicals, regulating most microbes to the industrial waste bin. I mean, this is like, right? Plastics. I mean, I was laughing at it because it's like, it seems ridiculous, but then you think about everything that's coming from that, right? You got the huge expansion into fossil fuels, the mining that happens there, and then you have the plastic boom, right? Everything starts being made from plastic. You know, it's just, yeah, it is what a sad time. It's hard to imagine, though, so much of our world without that happening. You know, even how I'm speaking to you now, you know, it has everything to do with silicon chips, and it'd be a different world if plastics weren't invented. That's for sure. But not all. Today, yeast and Escherichia coli are widely used to produce a range of commodity chemicals, including ethanol for fuel and compounds used to make pharmaceuticals and plastics. But these industrial workhorses come with their own environmental footprint, because the sugars and starches they ferment come from crops like corn and sugarcane. By 2026, corn grown to make ethanol in the United States will take up to 19% of the nation's farmland. Oh my gosh. Why? I mean, this is a weird issue too, because there's the bill that was passed by President Clinton that requires us to actually have 10% of our fuel be ethanol. So you have all these different starches and sugars coming from corn. Oh, that's so sad. Now, synthetic biologists are pushing to grow microbes on a more environmentally friendly diet using waste gases produced from industry, garbage dumps, and crop production, says Michael Kopke, synthetic biology at Lanza Tech, a biotechnology company. In the 1990s, researchers engineered Clostridium autoethangium, a bacterium originally enriched from rabbit feces. Whoa. To produce ethanol from hydrogen gas and carbon monoxide. What is this? In June 2018, LanzaTech opened its first production plant that used the bacterium to make ethanol from a steel mill's waste gas. A mix of mostly carbon dioxide and hydrogen gas would otherwise be vented into the atmosphere. The company now uses this technology to make 90,000 tons of ethanol per year. Jeez. Wow. Still, the bacterium has never matched the versatility of E. coli or yeast. Up to now, engineering Clostridium has been just hard, says Michael Jewett, in synthetic biology at Northwestern University. The bacteria are slow growing and they die from exposure to oxygen. In addition, researchers have few tailored gene edited tools to alter the microbe's metabolism. That's interesting. I mean, you would think that all the gene editing tools would be able to edit it. Why? How do they? Is it just tailored for that one organism? I don't understand. Not anymore. I love that. Jewett, Kopke, and other colleagues use a multi-step strategy to coax C. auto into churning out acetone and isopropanol, a colorless liquid found in disinfectants and cleaners. They started with a collection of 272 sugar-eating Clostridium strains maintained by retired South African microbiologist David Jones. The research team broke apart the bacterial cells and mined them for enzymes that appeared to play a role in the acetone biosynthesis. They used proteomics in order to see which ones were able to do it. I wonder if they had predicted which ones would be better, or if they just took all of them and then used like kind of an erased. Nope, they sequenced the genes for 30 of these enzymes and inserted combinations of them into C. auto via gene delivery vehicles like plasmids. Okay, so they're using like a directed evolution approach. Ultimately, Jewett, Kopke, and other colleagues generated and screened 247 genetic variants of the bacteria to find the best chemical producers. The resulting report out today in Nature Biotechnology are C. auto strains that continuously convert steel waste gases into enough acetone and IPA to make them viable candidates for large-scale commercial production. That is so cool. Yeah, directed evolution. If you can use nature's diversity and then screen that diversity, then we could try to create a product that is just better. Jewett expects that Jewett expects the same process could make the bacteria produce a variety of other chemicals such as butanol, used in varnishes, and propanediol found in cosmetics. Scown adds that novel synthetic biology techniques would also help transform other recalcitrant microbes into industrial workforces. We're just scratching the surface in terms of other microbial hosts that might be viable in the future. I mean, considering that 99% of microbes are not transformable, not able to be genetically modified, or even cultured in the lab, I think that there's a huge host of tons of species, millions of species of microbes, to be honest, that could actually make a big difference in the future that we have. The advance could also open up the door to genetic engineering microbes capable of feeding on other gases, such as those produced by municipal solid waste and agricultural debris. And if, like C. auto, these microbes are use more carbon in making their products than release into the atmosphere, industrial chemical production may someday transform from a greenhouse gas rogue into a champion. Well, let's check out this paper. Carbon negative production of acetone and isopropanol by gas fermentation at industrial pilot scale. Wow. Many industrial chemicals that are produced from fossil resources could be manufactured more sustainably through fermentation. Here we describe the development of a carbon negative fermentation route to producing the industrial important chemicals acetone and isopropanol from abundant, low-cost waste gas feedstocks such as industrial emissions and syngas. Using a combinatorial pathway library approach. Okay. So they use different parts, combine them together, and the library is all the different types of mutation and the different types of pathways combining together in order to make that product. We first mined a historical industrial strain collection from superior enzymes that we use to engineer the autotrophic acetogen, Clostridium, autoethanogenome. Next, we used omics analysis, kinetic modeling, and cell-free prototyping to optimize flux. Finally, we scaled up our optimized strains from continuous production at rates of 3 grams per liter per hour and 90% selectivity. Life cycle analysis confirmed a negative carbon footprint for the products. That's incredible. Unlike traditional production processes, which result in the release of greenhouse gases, our process fixes carbon. These results show that engineered acetogens enable sustainable, high-efficiency, high-selectivity chemical production. We expect that our approach can be readily adapted to a wide range of commodity chemicals. That's incredible. So the pathway optimization, the genome mining of everything, combining multiple of these. So they're different, multiple pathways with multiple different types of mutations in each pathway, each probed on each of, with every combination. That's pretty incredible. How are they able to have such a high rate of library efficiency going through a majority of their library if they didn't have those genetic engineering tools like they needed before? Cell optimization, the cell-free prototyping, omics, metabolic modeling. So that's to make sure that they're making what they're thinking. The cell-free prototyping is an interesting thing because they're just looking at the products of each of these pathways, I guess. And then they're scaling up to the process optimization, the fermentation and development, scale up, and then finally the life cycle analysis. Wow. Look at those, the acetone. They're all pretty good. Above the reference strains. That's incredible. Wow. That is really cool. Strain optimization, so you have the different pathways working in order to make the products. How did they know that each of their pathways, mutants, and combinations were going to be making their desired product? I guess that's how they're making the mutations here, and then they're doing that here. They're looking at the different pathways to see what's being made and making sure that each of them are being made along that way, that biochemistry. Because when you have so many different pathways here, you could have a lot of the resources being made and shunted towards lactate or something at the end here. And that's not what you would want. You don't want like lactate being where all your energy is going. So you'd have some type of flux monitoring. How are they doing that? Process optimization, and finally the life cycle analysis. So there you go. That's it. Microbes are the future. There's no stopping it. It's going to happen. Lou, 2022.